This was a fascinating concept when it was presented to us at Roswell Park. The company is SYNCAR-009. What this is basically, it's a multi-centered phase 1 study. It's dose escalation that we've reported on now and some safety and very preliminary efficacy data. But basically, this is a axi-cell-like construct, CD19 autologous CAR T that's been modified with an orthogonal IL-2 receptor and a corresponding orthogonal IL-2. What this is essentially is a modified IL-2 that is biologically inert, actually, and our study showed that part at least, that this mechanism and that drug work. There were no side effects of the safety lead-in, with that particular modified IL-2, as you would get, say, with traditional IL-2 with flu-like symptoms, et cetera. This study is the first of its kind, to my knowledge, to be driven by this kind of platform. And basically, you'll see T cells, whereas traditional CAR T-cell therapies will have T-cell kinetics where they will rapidly expand and proliferate, peak with corresponding toxicities like CRS and ICANS, cytokine release syndrome, and neurotoxicities, frankly. This study showed some peak, but actually, whereas normal CAR T-cell therapies would degrade and decay over time, you see the T-cells fizzle out over usually months. This platform has actually shown a stabilization of those T-cells, and it's allowed persistence and stemness through the treatment. So it's really cool. Efficacy for marginal zone lymphoma was excellent. For diffuse large B-cell, just like only a handful of patients for each was excellent as well. There's also a really cool extra cohort where there's no lymphodepletion being given as well. It's the Arm B of the study, trying to allow those T cells to expand in the body without chemo for patients who have, say, in next studies received CAR T-cell therapies or have been heavily pretreated with cytopenias. So we think it's a very cool platform. It needs a little bit more work and data, and they're generating that. It looks very safe, very, very effective for marginal zone lymphoma, large cell lymphoma. As far as my subniche in CLL, it's a little less effective, but it was still in dose escalation with some very nasty high risk and bulky disease. So it's one of the higher risk populations for cell therapy, period, I think. So more work is needed on the tumoral microenvironments. That's going to be my passion. I've encouraged them to look at some of our sequential biopsies for the patients who have progressed, unfortunately, with CLL to see why that treatment failed them, frankly, so we can make things better and hopefully with round 2 make it better for patients and more effective and still very safe and tolerable.